Alright everybody, welcome back to 303. This is Lab 2. Before we begin the lecture course, just for the online students, I wanted to quickly mention, in your notebook you have a list in one of the early pages. You have a table of every single assignment that's going to be assigned and also when it is due. Do keep in mind that the assignment numbers do not correlate with the lab numbers. So last week we had assignments 1 and 2 assigned, and they were due by this week, I believe for you guys online it should be Thursdays. Is that right, Max? Yeah, Thursdays. So make sure you submit your assignments before then. And again, just do keep in mind that the important thing is that the assignment numbers do not correlate with the lab numbers, so keep that in mind. So, easy lab today, pretty straightforward. So everything, I guess, is submitted online now due to the change, just to minimize risk of COVID, I'm assuming. So make sure you guys submit your assignments online. So all right, we're going to be going over symptomology and bacteriology today, and we're also going to be looking at Koch's postulates. So even though we've been talking about plant pathology, we haven't really defined it yet. So simply put, plant pathology is the study of plant pathogens or diseases in plants more broadly. And this involves many factors. You know, we can talk about host factors, we can talk about resistance, we can talk about the pathogens themselves. We can talk about that all day, but one of the more generally accepted ideas of those broad categories for plant pathology would be the concept of the disease triangle, in which we take a look at not only the host and the pathogen factors, but also the environment, because the environment plays a very large role into how much disease you see. When taking a look at animal pathology, and you typically only look at the host and then the pathogen itself, but since plants are unable to escape their environment like animals can, they have to deal with the environment and whether it's beneficial or detrimental to that relationship that they have. So in order for there to be disease, though, you have to have a susceptible host, meaning a host that is capable of being infected and have disease and be diseased by the pathogen. You need a virulent pathogen, virulent meaning it's able to produce that disease. And the environment needs to be conducive to that disease. So if you have an environment that is very high in temperature, you're not going to get a lot of good spore germination of certain fungi, and so you won't get infection. But if the environment is very cool and very humid, then you will get a lot of that germination of the spores, and you'll see consequently lots of disease in your fields. So all three play a very large role, very important role in plant pathology. Today, though, we're going to be looking at disease and symptoms. So here we have a Webster's definition of disease. It's pretty broad, and here on the right we have a picture of southern leaf blight of maize, which is a disease symptom of Cochlevolis heterostrophuson. Probably mispronounced that. But we have a more strict definition for plant disease. But before we get into that, we're going to look at what actually causes the diseases. Now there's, we call these agents that cause disease. We have biotic factors, biotic meaning life, bio meaning life, I should say. So these would be the pathogens themselves. So for example, fungi, nematodes, viruses, and even parasitic plants. And for the non-living agents, we would call these the abiotic agents. These would somewhat consider them to cause disease as well. These would be like having too much light or too little light, if it's too hot or too cold, as well as what chemicals are present in the soil or in the environment. You also have salt stress as well. And one thing I do want to point out, this is the definition of the pathogens and biotic factors that we're, abiotic factors that we'll be using in this course. However, do know that there is some argument between plant pathologists on whether a non-living agent can actually be a cause of disease, or as you can see here, we have viruses listed as a biotic pathogen, but some scientists don't even consider viruses to be a living organism. So for the purposes of this course, this is what we'll be asking you, or what is a biotic pathogen, or what is an abiotic or non-living pathogen. But do know that there are some discrepancies in that. So disease itself, in this course we're going to be going off of Agrios, who wrote the textbook, for his definition of disease, which is the response of plant cells or tissue to pathogenic agents, which negatively affects the form, function, or integrity of the plant. And you can see that's also kind of a very broad definition of what it is. But in order for a disease to be established, there needs to be both signs and symptoms, although you can't always see signs. There's always going to be symptoms. You know, you're always going to be able to tell if a plant is diseased or not, if it's displaying symptoms. So, you know, you guys have seen all this before, and especially with Sheila's presentation. So simply put, a sign. We'll go over what a sign is first. A sign is evidence of the pathogen itself. So, you know, by seeing fungi on a plant, you can say, oh, that's a sign of disease, or you can see spores of it, or something left behind, some remnant of the pathogen itself. Whereas a symptom is the plant's response to that pathogen. So that's where the distinction comes in. You only see a symptom of a plant once it's been challenged by that pathogen. So symptoms would be something that the plant is displaying. Here we have another example of what a sign would be. For example, on these grapes, you can see all this kind of fuzzy looking powdery stuff on there. That's all mold, so that's all some fungus on it. That would be a sign. You would be able to look at these spores underneath the microscope, or the fruiting structures, I should say, and definitively tell, hey, this is, you know, X pathogen. And that's how we're able to diagnose this disease. You know, part of being a plant pathologist, again, as Sheila was mentioning, was properly diagnosing plants with whatever disease that they have. And by being able to see the actual pathogen itself, well, that really helps out in what is it? Sorry, I've lost my train of thought. That would be a clear. What's the word I'm looking for, Max?
For diagnostic?
Diagnostic, thank you. Let's cut that part out of the video. So, if you can see the pathogen itself, you can clearly diagnose the disease. Symptoms, though, are a little more trickier because different pathogens can elicit the same symptoms in a plant. Although you don't always see signs, though, that's why you have to go off of the symptoms. So again, symptoms are just the response to the plant's response to being infected by the pathogen. So because of that, we're going to be looking at symptomology for today's lab. We're going to be able to, hopefully by the end of today, you'll be able to determine whether you're looking at a sign or a symptom of the infection. We're going to be doing a little bit of bacteriology, and we're also going to be continuing with last week's Koch's postulates. So here are the four general categories of symptoms. I'm not going to go over every single one of them because there's, even in these broad categories, there's still a lot more. But it's basically death or disintegration of the cells. We would call that necrosis, you know, death of the cells themselves. That would be similar to what we saw in last week's potatoes, where you see the cells actually start to become black and rot. You also have discoloration. The more common symptom that you see in many pathogens is chlorosis, which means that the leaves themselves, they start to turn yellow, and that's a sign of stress. That's a very broad symptom, though, so you can't really pinpoint or diagnose a disease based off of just chlorosis. And then there's, of course, other types of discolorations as well. We have wilt. Wilting, you can see some of the tomatoes here, tomato plants that we have set up are wilting. So some wilting is caused by pathogens when they start to block and clog the xylem or the phloem inside of the plant cells, so the plant body, I should say. And then you can also have abnormal growth and development. And this happens when a bacteria, specifically the agrobacterium, it infects a plant and it causes, it tricks the plant into creating more and more nutrients and starting to store them in a local space so that the bacterium can take advantage and start to reproduce in that. And I'll show you what that looks like in just a second. But we also have galls and crinkling. Just wanted to make note for today's lab for the online students. We'll be taking images, we'll be taking pictures of these plants and you know, some of the signs and maybe symptoms that you'll see. And we'll be uploading them, and you guys can look at that online and write down any symptoms or signs that you see. For you guys here that are in person, we'll be moving around a bit. As you can see, you got a little bit of something different at each table. And we want you to be very specific in how you name the symptoms that you see, because you know, just writing down discoloration is too general. So make sure you guys take very specific and detailed notes on that. So let's see, signs. Again, signs are the best way of diagnosing a disease, one of the best ways. All right, so and here we have some examples between different signs and symptoms. Now, as mentioning earlier, one of the bacteria that causes wilting in plants is because they start to clog up the xylem, which is where all the water flows in the plant. So the way they diagnose this is they'll cut a branch, or they'll cut a section of the branch of the infected plant, what is suspected to be infected, and they'll put it in water. And you get what's called this bacterial oozing, or bacterial streaming, I should say. And this is actually the actual bacteria that is streaming out of the plant. So would this be a sign or a symptom of disease? That would be a sign, exactly, because you're seeing the bacteria itself. Now, I cannot ascertain whether this, you know, necrotic region is caused by the bacteria itself, but it likely is. But this black spot, would this be a sign or a symptom? Yeah, if it was caused by that bacteria, it would be a symptom, because this is the plant's response to being infected by the bacteria. Over here we have this, you guys have probably seen some of the trees on campus here with these huge growths, especially near the surface of the ground. Would this be a sign or a symptom? It's basically a huge tumor. This would be a symptom, right? If you were to stab in here and, you know, draw a knife out and with a knife and then put that knife on one of these plates, you'd probably get agrobacterium growing. Because again, what the agrobacterium does is they start to produce all these hormones that cause the plant to grow bigger and bigger without differentiating, kind of like a tumor how you see in humans. Except the bacteria they take advantage of the plant by consuming all these nutrients that the plant produces. So it's a pretty cool mechanism, and you see some of your tomatoes have that as well. These are also called galls. Over here we have, it looks like a mosaic. It's kind of a grainy picture. But would this be a sign or a symptom on the leaf? I assume it's, let's just say, because it's so blurry, let's just say it's sclerosis or mosaic. Would that be a sign or a symptom? It would be a symptom, right. This, the mosaic and ring spot is mostly caused by viruses. With viruses, you never really see signs unless you're looking at the virus itself under an electron microscope, because you know, viruses are so tiny. You can see vectors of the virus, vectors being what transport the virus from plant to plant. Those tend to be insects, but they're not always. However, you really have to go off of symptoms for viruses, and a lot of it goes down to molecular testing. Some viruses do produce very specific symptoms in their hosts, and you can clearly tell which virus it is based off of that. But for many viruses, it's best to do molecular testing to ascertain which virus it is. All right, so that is the end of the lecture part.